Hello and welcome everyone to another demonstration video from Biotecnika. In today's demonstration, we'll look at a tool called as, or basically a web server called as your Gene Ontology. Okay, so in this particular web server, we'll look at how we can do the gene enrichment analysis or basically pathway enrichment analysis. Apart from that, we'll also look at how to do gene ontology. So what is gene ontology in the first place? So basically this is a computational method of storing the things that how a particular gene product or basically its function can be divided into multiple components of us at cellular level. Okay, so it involves three categories basically, like your biological process, cellular component, and your molecular function. Okay, so we'll look at both of those things. Okay, that is your enrichment analysis and your gene ontology using the same server. Okay, so without further ado, so let's get started. Okay, so in your regular search engine, whatever search engine you are using, you just have to type gene ontology. Okay, so here you have it. Okay, so gene ontology resource is the full name. So if you go inside this, okay, so you have this particular home page over here. Okay, so if you want anything to like you want to know about the annotations, you want to know about ontology, you want to go to help, frequently asked questions, so that you can obviously go on to. Okay, so we'll take some example in today's demonstration and we will see that how the things works. Okay, so first things first, we'll look at gene ontology terms first. So that is your we will take a gene for example. Okay, so you can take any gene for example as such. Okay, so I will take a very known gene only, so BRCA1, BRCA1, so that is responsible for your breast cancer. Okay, so if you directly want to go on to ontology terms, you can always click on this one. Okay, so you can click on ontology and then search, but as you might be new to this particular web server, let me show you what are the options that it gives. Okay, so once I click on this one, okay, so yeah, so we'll be redirected to the page, and here it is giving us an overview of all the results that it has got. Okay, so first thing is your ontology term, then your gene and gene product, then your annotation. Okay, so obviously here we are interested in the ontology term. Okay, so we'll go on to ontology. Okay, and we'll see the results over here. Here are the results. Okay, so if you want to use some more filters, you can add them, or if you want to like omit any of them, so you can simply click on this particular minus and that particular thing will be filtered out. Okay, so apart from that, gene ontology sources, if you want to add any filters, that you can do. So these are basically filters in the left hand side of yours. Okay, so we'll go on to this. So let's go on to the first search result. Here you can see the ontology terms are already given, that it is a cellular component or like biological process it is involved in, or which molecular function it is involved in likewise. Okay, so this is the complex, and here you have the what do you say, your detailed information of the same. Okay, you can follow the links also if you want to. Okay, and this is the, you can see the gene product, what is the like from where it is having the evidence, and the protein product, what it codes for. If you want to know the, what do you say, the hierarchy of the things, you can see the Panther family classification is also given over here. Breast cancer type 1 susceptibility protein BRCA1. Okay, so if you want the evidence, you can always go to UniProt. So UniProt is the protein database. So let's for example get onto that just to show you. Okay, so this is the UniProt ID of the same. Okay, so this is the BRCA1 gene. You can go and search for the sequence and likewise. Okay, yeah, but the main thing that it gives is the classification of the thing, the particular gene into multiple terms that we are gonna see. Okay, apart from that, you can also see the hierarchical view. Okay, so if you go to graphical view, okay, and you go to quick GO, gene ontology, here you have the chart based representation or the workflow based representation of what are the cellular components. It divides it into, okay, positively regulated, negatively regulated, how like based on the process, function, and component. And these are the main three terms in which a GO term is classified. Okay, so that is the main part that you need to learn and understand. Okay, so here you have all the IDs also. So based on what is your research question, you can search much more. I'm just giving you the overview of this particular web server, what it caters to. Okay, So yeah, so that is it. Then you can also go to inferred tree view of the basically that particular hierarchical thing, then the neighborhoods of that based on the like distances. Okay, so evolutionary distance I'm talking about. Okay, then your mapping of the gene, multiple genes if you are adding, then you can also do mapping of the genes and likewise. Okay, yeah. So now let's go to the next part real quick, that is your gene enrichment analysis or GO enrichment analysis. What is this? Basically, here we will look at what are like how are the genes being expressed, or basically pathway enrichment analysis you can also call it as. Okay, just like what you do in KEGG. Okay, so likewise a similar thing you can do, but this will give you a detailed view based on GO term. What are the GO term? Here are the GO term, that is biological process, molecular function and cellular component. Okay, so you can select either of them if you want the results in like whichever process you want it in. Okay, apart from that you can obviously change the organism as well. Okay, so let's take some cluster of genes basically so P like PNPLA3 gene will be taking causing your NAFLD. Okay, non-alcoholic fatty liver disease. Then your GATA3 which is involved in your gastrointestinal anomalies. Okay, so then you can also have your cancer based gene, that is your BRCA1. Let's suppose we take BRCA1 again and BRCA2 also. Okay, so likewise you can accommodate multiple genes over here. Okay, you can also hit on example and they already have a plethora of genes, so that will be loaded and then you can hit on launch. Okay, so you have the Homo sapiens selected over here. Biological processes I want to know, or let's suppose I wanted to know molecular functions. Okay, so I can hit on launch. Okay, so once it is launched it will give me so it will ask that I'm not a robot. I will continue. Okay, and here you have the search terms. Okay, so depending upon the uniqueness of the thing, so you can see unique mapped IDs. Here you can see this many. Okay, from the reference list. Okay, and whatever we have uploaded, 4 of them are obviously 4 of them only we have uploaded, and the hits it can find in this particular results. Okay, so here are the results. Okay, and now obviously this particular representation that you see over here, that is the tabular representation, and you can, but this is not very easier to infer. So you can always go to your this particular kind of a representation. Okay, like you can do it a pie chart or a pathway enrichment kind of a chart. Okay, so multiple viewing options you will be having. Okay, bar plot also you can do. So here you can see, so what are the different components. It is like unclassified is also there. Obviously all of the things are not classified over here. Apart from that, the different classification that you can see, ATP dependent activity is connotated by this particular color. Okay, binding is connotated by this particular color, and likewise based on the color connotation you can see. Okay, so you can also like do a view of 125%. It is nothing much. It will just increase this particular view of yours. Okay, you can also go to a representation of bar chart or bar bar plot if you want. Okay, so apart from that, from this particular drop down menu you can change the particular ontology term also. Okay, so like we are currently in your molecular function, I can change it into cellular component. If I change it into cellular component, you can see so 2 basic cellular component, it divides it into. Okay, so obviously one is unclassified, so leave that. So these are the 2 main categories that it classifies into. Okay, so that is your cellular anatomical entry, and the second one is protein containing complex. Okay, so apart from that you can also go to, let's see about the third part also, that is your biological process. Here you will be getting much more results because multiple biological processes it would be involved in, that you can see over here. Okay, likewise. Okay, and you can also see the pathway enrichment part that I was talking about. Okay, so you can see what are the pathways all of these genes are involved in. Okay, so here you can see one is like very much prevalent. Okay, so here you can see that particular category. Okay, yeah, this one like shown in your pink. Okay, so all others are there. It is like so small that you can't see. Okay, but if you point your cursor towards here, or let's get a pie representation would be the right representation over here. Okay, here you can see the most of the part is your this one. Okay, connotated by a light pink over here. Okay, so apoptosis signaling pathway mostly. Okay, let's see. Oh no, this is unclassified, pardon. Okay, this is unclassified, and this is the thing that I was talking about, what it is involved in, you can see Huntington disease that I pointed into. Okay, so likewise there are multiple classification. Okay, so filter unclassified, that also you can do, because unclassified has no meaning for us, isn't it? So now you can see the representation. It is a better representation of what we want to see actually. Okay, so this is the representation that you can see now. Also, like same bar plot for the same. Okay, now you can see, because we have removed the unclassified one, so that is why it is now very much prevalently we can see all of the representation over here. Okay, and if you want to see what the which color connotation leads to what particular kind of a process, that you can follow from here. Okay, so that is all from my side as of today. Okay, so this is Pradyut Banerjee from Biotecnika signing off. Okay, so if you have any questions, comment down below, and if you like the video, please do share it with your peer ones who are in need and who wants to join this particular course from Biotecnika, that is your Bioinformatics Global Tools and Techniques. Okay, so that is all from my side. Bye bye, take care.